we've been discussing about the fact we have different types of lab investigation methods in medicine. And microbiology is just part of all of these investigations. Microbiology is just part of all of these investigations. So if we are talking more specifically about serological reactions, which we already discussed, and I would like to remind you that serological reaction is a reaction basically between antigen and antibody. So if the reaction between these two components happened, the reaction is called positive. If they didn't bind each other, reaction is negative. And also we can classify all the serological reactions in two main groups: sero-diagnosis and sero-identification. If we are talking about sero-diagnosis, we are talking about known what? We are using known antigens in order to find unknown antibodies. In this case, we are taking the blood from a patient because we are looking for unknown antibodies in the blood, and we are taking known antigens in the lab. We make reaction. If reaction happens, that is positive. And we have sero-identification. Sero-identification when we use known antibodies in order to identify unknown antigens. In first case, we are looking for antibodies. That means we check for immunity. In second case, we are looking for antigens. That means we are looking for a pathogen, because antigen is pathogen. Or antigen is a part of pathogen. So today we will discuss about one more classification of serological reactions. First of all, we can divide all of the serological reactions by other features. For example, by number of components we have in this reaction. So, all the serological reactions also by amount of components can be divided in two groups: double component and monocomponent. And if till now we discussed about the fact that basically serological reaction is reaction between two components, that is antigen and antibody, now how it can be monocomponent? It should be, all of them should be double component. Why it can be monocomponent? And now I will explain how it is possible. So if we start with double component, that is mostly classical and mostly clear for you, we can also divide them in two main groups: simple and complex. If we are talking about simple, there are some examples. Agglutination reaction. Agglutination reaction is when antigen and antibody are connecting to each other. They make this agglutinate. What means agglutination? What means glue? Sticking to each other. So agglutination is binding, is clumping to each other, making a huge complex that becomes visible. Before reaction, liquids are transparent. After reaction, liquids, we can see agglutination, so we can see visible particles because they become very big. They clump to each other. And these particles stop, they are making the solution to be non-transparent. They give turbidity to the solution. So we can see the positive reaction. We can have also indirect agglutination reaction. Also, here will participate more components, and I will describe it later. Precipitation reaction. What means precipitation? What means precipitation? Are you listening or reading some meteo prognosis? Did you hear about precipitations outside? Precipitations from atmosphere? What do we call precipitation in such case? Snow, rain. These are natural atmospheric precipitations. So what means precipitation? Falling down. When something falls down from the up. Also in forensic medicine, did you have it or you will have it? You're gonna have it, yes? In forensic medicine, you also will hear about precipitation. What does it mean? You will hear about death through precipitation. If a guy or anybody else is climbing the building and after that steps in the air, what happens? Precipitation. So if the building is high enough to produce death after such precipitation, in forensic medicine you will hear such definition as death through precipitation or falling down. It is also precipitation. In microbiology, we also have precipitation with some more positive consequences. Precipitation in the solution. So, if after a reaction, usually it happens in tube, we put some solutions with antigens, with antibodies. So if after this reaction something will fall down in the bottom of the tube, precipitation happened. What does it mean? That means that till now, all the components of the reaction wasn't that heavy to fall down and to connect into some agglutinates. But since they clump to each other, they become much heavier and they fall down in the bottom of the tube. So this reaction will be called precipitation reaction. If it is positive, it will happen. If it is negative, it doesn't happen, you see nothing. And also we have lattice agglutination test. Lattice agglutination test. Did you hear about lattice agglutination test? I guess some of you did, some of you didn't, but basically it is not that common for general population. Lattice agglutination test. Basically what we need to know about this. The reaction is similar as hemagglutination, with hemagglutination test. So clamping of erythrocytes. Because particles of latex, do you know the origin of latex? Which is the origin of latex? It is a natural organic substance produced by what? By a plant, kind of tree. So if you cut the tree a little bit, it will start eliminating a whitish juice. It is latex. So this liquid is used for production of gloves, of condoms, of any other products made of latex. So it is not a rubber. It is not artificial or synthetic product. It is a natural product. This is why you will never have, you can never have allergy for rubber, but you can have allergy for latex. This is what you probably heard, that some people, they have allergy for latex. This is because latex is a natural, it is a natural matter. It is not synthetic matter. Okay, so these small components of latex in the liquid state are looking similar by their structure with erythrocytes. So the main function of latex agglutination test is replacing of hemagglutination test. So we just save blood by using latex. These are simple double component serological reactions. Also, we can have complex serological reactions. For example, neutralization reaction, indirect hemagglutination reaction, reaction of hemagglutination inhibition, and immunofluorescent assay, the direct one. This is a complex. These are complex reactions, and these reactions will be double component. That means all the time in these reactions, will be present both antigen and antibody. Now we can talk about monocomponent reactions, in which will be present just one component, either antigen, either antibody, but just one component. For example, indirect immunofluorescent assay. For example, radioimmune assay, immunoenzymatic assay, and complement fixation assay. And if you will learn a little bit about these types of investigation, really you will find that there is present just one component of the antigen or antibody reaction. But this time, some extra substances will be used in order to identify these structures. So these are reactions with monocomponents. Now, if we are talking about neutralization reaction, that makes part of double component complex reaction. So it is based on the property of immune serum when it is added to a virus, for example, to block its ability to reproduce. Neutralization reaction. Very often, just by reading the name of serological reaction, you will understand partially or totally the meaning of the reaction. So if you can hear neutralization reaction, that means in this reaction will be neutralized ability of the virus to replicate, to reproduce. So in serological diagnosis, neutralization reaction is used to detect and increase in the titer of neutralizing antibodies to a specific or known antibodies of known virus that is present in blood. So by this neutralization reaction, we will observe, does our body produce enough antibodies against virus or not? If we are talking about hemagglutination reaction, that is also making part of of serological tests, hemagglutination reaction. So it is when blood cells are clumping to each other because of what? It is monocomponent reaction. Hemagglutination reaction is monocomponent reaction. When it happens, do you remember from virology, a lot of viruses, they have antigen that is called hemagglutinins. Do you remember? So most of viruses, you can detect them by two main antigens, hemagglutinins and neuraminidase. This is why when you are looking for some flu viruses, for example, their name will be based on H2 and 3 or H5 and 7, imaginary. What does it mean? H means hemagglutinins antigen, N means neuraminidase, and the number means number of modification or mutation of this antigen. And by knowing the number of mutation of each of them, we can choose the mostly specific treatment or mostly specific vaccine for this virus. So, what means antigen hemagglutinins? Hemagglutinins, by its name, you can understand that it has ability to attach to the red blood cells. So as soon as the virus will get in the bloodstream, as soon as virus will get in the bloodstream, since on its surface we have this antigen that is called hemagglutinins, it is able to attach to red blood cells. One virus can clump together more erythrocytes because of this antigen. Is it serological reaction? Yes, because it happens in blood. Is it double component reaction? No, because we have just virus, just antigen. So this reaction is monocomponent serological reaction that happens in blood. So if we have clumping of erythrocytes, the reaction of hemagglutination will be considered positive. That means in the blood we have virus. It will be monocomponent serological reaction. Now, if we will add in the blood some antibodies for this virus, what will happen? Hemagglutination inhibition reaction. It is double component serological test. Can you make difference? Did you get it? So, we add some antibodies against this specific virus. We add it in the blood, and the clumping will stop. This is why, if you remember, while pandemic, a lot of people were dying because of what? Because of thrombosis. Do you remember? A lot of people who've been infected with COVID, with coronavirus, have been dying because of thrombosis. Why? The presence of the virus in the blood produced in vivo, that means in the living body, this monocomponent serological reaction with clumping of erythrocytes. When they clump to each other, they produce thrombs. They block blood vessels, and thrombosis starts. So thrombocytes are starting making these clumped erythrocytes to make them more bind to each other. This is the process of thrombosis and of obturation of the blood vessels. As a result, you don't have proper bloodstream, blood circulation. As a result, decreasing of oxygen. As a result, necrosis of some tissues can happen. As a result, we have a death. While we've been talking about vaccination and a lot of people were saying, oh, it causes thrombosis. Come on. It prevents thrombosis indirectly. So, vaccine. So, vaccine prevented thrombosis caused by erythrocyte, virus. So these two reactions, hemagglutination and hemagglutination inhibition, are making part from different groups and can help you to detect presence of a pathogen in the bloodstream. Talking about complement fixation reaction. It is a complex reaction that occurs in two phases. So first of all, we take known antigen and apply this known antigen to unknown antibodies. That means as a specimen, we take from a patient a blood. So, we take a blood serum from patient to detect presence of what? Presence of antibodies. And this reaction is called, also, this part of reaction will be called serodiagnosis, because we detect unknown antibodies by using known antigens. Second reaction is called, will be called, or will be making part of sero-identification, because we take unknown antigen and known diagnostic immune serum. So, if, and this reaction is happening in two steps. So, if a specific complex is formed, reaction between antigen and antibody, if this reaction happens, so there is a complement. There is a complement that remains free. The complement is non-specific component of our immunity, if you remember, that is able to produce lysis of cell wall, lysis of erythrocytes, but it is not non-specific. It can attach to both pathogens and blood cells. So, if the reaction happens between antigen and antibody, the complement will destroy blood cells and we will have hemolysis. In the first part, and this hemolysis will occur just in presence of free complement that didn't bind to any pathogen or to any antigen. If specific complex is formed and the complement will be attached to this specific component, to this specific binding, hemolysis will not happen. And in this case, we will see that complement, indirectly we will understand that complement will destroy the pathogen. We cannot see that complement destroys pathogen, but we know that if it remains free and it doesn't attach to specific component, antigen and antibody, that means it will attach to erythrocytes. Reaction didn't happen and hemolysis will appear. If reaction doesn't happen and hemolysis doesn't happen, that means indirectly we understand that this complement destroyed the pathogen. Is that clear? Also, we have such reactions as passive hemagglutination test or indirect hemagglutination test. Which is the difference between direct and indirect hemagglutination test, or passive and active? Well, this specific passive hemagglutination test, it is that kind of test that detects presence of virus in the blood cell, in the bloodstream. Excuse me. Also, the immunofluorescent assay. Immunofluorescent assay the direct one, we told, is making part of monocomponent. So in this case, to the antibodies will also attached a fluorescent marker. So such reactions we will observe under the fluorescent microscope. And this marker, if it will be able to attach to the antibody, the reaction is positive and we will see the specific complex. If the attachment doesn't happen, we will not see any specific reaction. ELISA. What is ELISA? Enzyme-linked immunosorbate assay. Very good. Everyone knows to tell what it means, but who knows to explain what happens there? Usually for the test that's enough, yes? So, still, it is one of the bunch of immunological tests that helps to detect specific antigens. What does it mean? Is it seroidentification or serodiagnosis? Seroidentification, because it helps to detect specific antigens. That means this technique will be used for diagnosis, not for immunity checkup, right? So, the basic principle of ELISA in the specific binding is of the first antibody to the target. If the target molecule is a protein, when it is purified, and it is usually used to obtain antibodies, it will help it will help the target with antigen to be bind. So, what makes different ELISA from simple serological test? Simple serological test, you put together some reagents, and reaction either happens, either it doesn't happen. But first of all, ELISA is a reaction that is made by a machine, and the result is much more precise, because before we'll see the result, the reaction will pass through more stages that will purify all the components of the reaction. And when we are talking about diagnostic reactions, also we are talking very often about such reaction as PCR. What means PCR? Polymerase chain reaction. Is it serodiagnosis or seroidentification? Huh? What is PCR? Serodiagnosis or seroidentification? Huh? Louder.
Both are wrong answers.
Both are wrong answers. Yes? Why?
It deals with DNA amplification.
It is DNA amplification. Very good. I already asked you this question, right? Well, we can misuse PCR test with some serological reactions. Why? Because, as well as you know, the sample for PCR is taken in the same way how it is taken for seroidentification. And we will be looking for the pathogen. In case of seroidentification, we are looking for pathogen or for its particles. When we are talking about PCR, we are looking for detecting of a pathogen through amplification of its genetical material. So if it is also a reaction that is performed by a special device that artificially reproduces the genetical material of the pathogen. If in the initial sample we have any pathogens, after amplification or after multiplication, reproduction of the pathogen's DNA or RNA, depends which pathogen we have. If we'll have any positive result, that means a lot of genetical material of the pathogen. That means in the beginning we had something. If not, we will not. So if the reaction is negative, that means in the initial sample we didn't have any pathogen.